Hello and welcome to the webinar, A New Approach to Using TMS in Depression, hosted by the National Institute of Mental Health. My name is Kayleen DeHutt. I'm a social worker here at the National Institute of Mental Health in Bethesda, Maryland. Thank you so much for joining us today. Our presenter today is Dr. Bruce Luber. He is an experimental psychologist and neuroscientist who received his Ph.D. from New York University, where he used MEG to study the brain basics to spatial attention. Over the last 23 years as a professor at Columbia and Duke Universities, and presently as a staff scientist at NIH in Bethesda, Maryland, he has conducted brain stimulation research into the neurophysiological basics of cognition, memory, and perception, developing TMS paradigms in conjunction with brain imaging and EEG that target and modify specific brain networks in the hopes of reducing cognitive and memory deficits in aging and in psychiatric and neurological disease. Over the past 3 decades, he has also had a strong role in developing the use of ECT, MST, and TMS in the treatment of depression.
Okay, thanks for joining me here. I'd like to discuss what I consider a very exciting new approach to using TMS, transcranial magnetic stimulation, in alleviating depression. In terms of an outline for the talk, I'd like to talk a little bit about depression strictly as a brain disorder, and then talk a little bit about TMS and the use of TMS in treating depression, and then get into a new way we have of using TMS to interact with the brain and what I think create a much more efficacious means of treating depression, and talk about some of our studies along those lines. So depression itself is a very common mental disorder. It globally affects more than 300 million people. And there's just a great need for more effective treatments for depression. It's become clear that drugs that are used as antidepressants work, but only to a certain degree. There's a lot of side effects. And about 1/3 of people are not actually able to be treated by the antidepressants. TMS came along over the last few decades, and it was recently, in 2008, approved by the FDA for use in treatment-resistant depression. TMS itself is, for those who don't know it yet, we essentially take a magnetic coil, essentially loopings of wires, put it very close to the head, and send a very, very brief electrical pulse, a very strong electrical pulse through the coil. That creates a magnetic field that in turn induces currents in the brain. These currents aren't very large. They're kind of on the order of the currents that are already in there that neurons are using, but they're strong enough to actually affect neuronal activity and create things like action potentials, et cetera, and they're thus able to modulate what's going on in the brain. So TMS, as I said, was approved in 2008 by the FDA for treatment. This is based on some very large trials. We were a part of those trials ourselves at Columbia, just to compare the effectiveness of TMS compared to, say, placebo, actually sham TMS. And what you can see here is that TMS given daily on a weekday basis for about 37 minutes at a time every day for 6 weeks. Oh, there it goes. Wait, that did not want that to happen. You can see that there's a growing effect of TMS in terms of the rate of response and the rate of remission over a 6 weeks period. So the FDA approved essentially a 6-week period of using TMS for depression. The problem is you can also see that the actual response and remission rates aren't that high. They're on for these very large clinical trials, they were on the order of 24%, 25% response rates and 17 or so % remission rates. We've gotten a little better than that since 2007, but nonetheless, these still are not as large as we would like. And there may be reasons for that. One very important reason is that this is basically technology that was developed in the 90s in terms of the paradigms that were used for depression. And so the way that got approved depression was to find a place in the motor cortex that actually will stimulate a muscle in the hand. And you essentially move 5 centimeters forward, and that's where you stimulate it. The target for TMS was supposed to be dorsolateral prefrontal cortex. Place we wanted to stimulate was forward from that, anterior from that, in dorsolateral prefrontal cortex, and it wasn't quite even reaching that on most people. There are much better ways now of finding that spot than just using a scalp location that have been developed since then. And so we can use brain imaging, we can use fMRI, we can use resting state connectivity, DTI, diffusion tensor imaging. We can use neuronavigation to co-register people's MRIs with their actual 3D situation that where they're sitting in a chair. We can use electric field modeling. We can use much better means of coil holding. All these techniques have been developed since the original means of the original paradigm in depression was developed. So we have better ways of attempting to stimulate actual brain targets. The other aspect is when you stimulate with TMS, you're stimulating not just the site below the scalp area where the coil is, you're stimulating everything that cortical area that's being stimulated is attached to in the brain as well, so other networks. And what we have also developed, and especially at the NIMH, is an idea of using the knowledge that we have in neuroscience that we've been developing to actively decide on work and engage neural networks that have to do with symptoms of things like depression. This is called an RDoC approach, the Research Domain Criteria approach. So what we really want to do is engage specific neural networks that have something to do with depression. And again, this is a far cry from the original idea of let's just stimulate over dorsolateral prefrontal cortex and hope for the best. This is trying to actively engage and modulate specific neural networks using TMS. And in depression, we've already identified, we and many groups have identified certain neural networks that seem to have a lot to do with depression itself. Cognitive control networks that have mostly to do with cortical areas that have to do with how you decide on goals and decide on approach and avoidance and things that properties that are very much related to what goes wrong in depression. Things like the default mode network, another set of areas that seem to have a lot to do with thinking about your situation, ruminating, and then other areas that are even more primitive that have to do with valence, that have to do with what is good and what is bad, more primitive emotional systems, part of the limbic system, for instance, that have to do with, again, with emotional regulation. So there are a number of target networks that we can approach using TMS in this more scientific way. So another thing that we have developed in the meantime is an idea of functional targeting. Okay, not just targeting spatially, but what we have found is that most of the problems in terms of efficacy with TMS itself has to do with the fact that when you stimulate a part of the brain, there's two factors involved. One is the actual stimulation by the TMS, the external stimulation. The other is what's going on in the brain at the time when you stimulate. The brain's always active. It's always variable in what it's doing. And to get a little bit more control over the brain itself when you stimulate may be more efficacious, and we have reasons to think that. For instance, just in a very, very simple way, if you stimulate motor cortex, you will create effects in muscle And you can actually see that by measuring electromyograms of that muscle. When you actively think about moving that muscle, you increase the response. Okay? So when you actively use that area, when processing is occurring in that area, you create a bigger response to the TMS. We want to use that. And so, for instance, you can actually, we're hoping to use it in a way that was first proposed by Hebb more than half a century ago now, in terms of firing together and wiring together. Okay, so when you stimulate with TMS, all right, and create action potentials in that sense, but also are creating action potentials internally by mentally controlling that function, what you're doing in some sense. We hope that there's kind of a synergy there that modulates synapses as well in a much more efficient way than if you were just stimulating, and hoping for the best with the TMS, which is what's happening now. So it's not a very, very simple paradigm that demonstrates this. This is, again, in motor cortex. It's called paired associate stimulation, or PAS. On the far left, you can see that there's an EMG response to a certain stimulation over motor cortex. If I then, in the middle part of that diagram, if I then stimulate, okay, a muscle, the very muscle I'm trying to affect, or rather the nerve associated with that muscle, a little bit before I stimulate with a TMS, so that both the arrival of the information from that stimulated part of the muscle in the hand arrives in the brain. At the same time I stimulate with a TMS, I'm creating two forms of stimulation in that area. When I do those pairings, those paired associates, a number of times, and then I look at the response to TMS alone, you can see that it's enhanced. Okay, and that's the following diagram in the left diagram. And so that enhancement is a form, we believe, of long-term potentiation of synapses. And it's been demonstrated now that PAS kind of stimulation increases the effectiveness of learning tasks. And that's what the diagram on the right is showing. It's showing that there's increased accuracy with the PAS as opposed to not using the PAS or using it in a way that would actually be detrimental. So it really looks like that this is a way of being able to functionally control the state of the brain when you're stimulating and create a much more efficacious stimulus. And so we generalize that, and we call it cognitive paired associate stimulation, or CPAS, as a more effective approach of functional targeting. And we've incorporated that into a new way of doing TMS and using it in terms of cognitive deficits and in terms of psychiatric illness. And that includes this sort of targeting where we're actually trying to engage a specific functional network in the brain and use functional targeting to make that network active while we're stimulating. So it's a multimodal approach to stimulation, and we'll do this over repeated sessions to create a cumulative effect, over time, so that the changes that we make become long-lasting. So just to demonstrate this is some work we've did over the last 15 years to remediate a cognitive deficit, specifically in this case, a working memory deficit. And so we used a working memory task, a very simple one. It's a Sternberg task. And it's basically, I show you a letter or a set of letters, wait a little bit, and then show you another letter and just ask you, was that letter one that you just saw or not? So very, very, very simple idea. And you answer yes or no as fast as you can. So reaction time test. You can see there's two trials shown in front of you. With this one, the first trial is in H, and you see that the test letter is in H, so you'd answer yes to that. The next is 6 letters, and the test letter is a P, and P is not one of those 6 letters, so you'd answer a no. So very, very simple task. It's a very well understood and well controlled task, and we know a lot about where in the brain this task is being processed. And so we actually, at first said, okay, well, how, as a very first step, can we show that we can enhance using TMS working memory in this task? And you have to figure out what kind of stimulation to use. So, for instance, what is the frequency of a train of TMS stimuli that would work the best? Where in the brain we should use it? When during the task we should apply it? Okay, so we tried all those things. We tried various combinations, and what we found was that 5 hertz, so 5 stimulations per second of TMS, given during the retention period, that's the period between when we show you the letters and we're testing you on it, and in this case, to the midline parietal area, okay, which lit up in an fMRI recorded during this particular task. So we're stimulating in that network that medium parietal area is part of, worked the best, so that we actually sped people up without changing their accuracy. We actually made people faster at the task, just healthy volunteers, when we stimulated in this way. So then we said, let's create a deficit, a working memory deficit in people, a temporary deficit. One great way to do that is with sleep deprivation. We'll basically keep you up for 2 days. You will slow down in all your responses, and you will space out a bit. You'll have little micro sleeps where you won't even answer the question. You won't even answer the question. We have to kind of wake you up and have you keep going. So we actually create a deficit in people, and we found a network in the brain that reduced its activity over the 2-day sleep deprivation period, and the more the activity was reduced, the worse people got at the task. Okay, pre-post sleep deprivation. So we actually have a network. We have a target to use, and we have a little picture on the upper left of that network on the left-hand side of the head, and the lateral occipital cortex. You can see is one of the areas that we stimulate. We stimulate 3 different areas. Those hexagons are showing. Those hexagons, octagons, are showing 3 different targets that we used. The top one is actually that medial parietal area, which also was lit up in this part of the network, but you can't see it because it's in the middle from this angle. And we stimulated right outside, 1 centimeter away, right outside the network in the lower right there. So we stimulated 3 different places in each person who was sleep deprived. And what we found was when we stimulated inside the network, and you can see that in the right side graph, in terms of reaction time, we actually sped them up closer to where they would have been if they weren't sleep deprived. We also found that in the lower left graph that the amount that we sped you up, the amount that TMS actually helped you, was correlated with the amount that you went down as a subject in the network activation. So the more your network was deactivated by sleep deprivation, the more TMS helped you. So it's a nice brain behavior relationship. So we could show acutely that we could make people better in this deficit. What we really wanted, though, was something that was more long-term. So we actually applied our CPAS approach in the next experiment, and we stimulated that lateral occipital cortical area for the 2-day period while people were being sleep deprived, and they were doing the task while they were getting the TMS. So, again, we were doing that simultaneous, concurrent, Hebbian-like approach, okay? And we were trying to do it in a cumulative way where we did it over 4 different sessions. Each one lasted an hour, while people were getting sleep deprived. And then we tested them. So the last time they got that concurrent TMS task activation was the previous day. And then the next day, while they're still sleep deprived, we actually tested them on the task itself without TMS, and found that they, while people that got sham TMS, got fake TMS, were slowed down, acted pretty much like they were sleep deprived in the task. People that got sham TMS were slowed down. If you're above this line, you're slower, okay? If you're below the line, you're faster. People got active TMS that were sleep deprived actually looked like people that were just control subjects that were not sleep deprived. So they actually sped up a little bit, mostly probably due to practice, but nonetheless, not showing any deficit. And they showed, this is people that were sleep deprived, so people who were sleep deprived and had sham TMS showed many more lapses, okay? sleep deprivation, which is what people normally show with the deficit. But people that got active TMS did not show those lapses, or showed very minimally. So we effectively got rid of, in this one case, we specifically aimed at getting rid of the memory deficit, working memory deficit, due to sleep deprivation, and we did succeed. People that got the TMS in the CPAS paradigm showed no effect of sleep deprivation, whereas people that did not get active TMS did. And we were actually able to show that the coil was here, and right underneath the coil was the only place we saw a difference. There was, in fMRI, pre-post sleep deprivation. Namely, there was a more activated cortex below, in lateral occipital complex cortex, right below where the coil was. So we actually created a change in the brain that was long-lasting in the sense of another day. Once people are allowed to go back to sleep and everything, that all this deficit goes away. But we were able to show that at least 24 hours later, we had still affected this network. So we were very hopeful at this point that we had a paradigm now that could help people that are sleep deprived, that have a working memory deficit. And the National Institute on Aging actually funded us after that to work on elderly people, all people that once they hit about 70 years or so have some working memory deficit. That's just a normal thing. We're actually already able to show that stimulating, for instance, lateral occipital cortex in these people improves their working memory. We compared them to younger people as well. This is soon to be published data. So getting back to depression itself, what we want then is to apply this CPAS paradigm to people with depression. So first of all, we need network, and there's a number of networks that are potential to be used in depression to engage with. We prefer looking for one that's in the cortex because that's easier to reach with TMS. And in fact, dorsolateral prefrontal cortex is part of a network that seems to have to do with emotional regulation. And so we decided to let's aim at that particular network to engage. And more than that, we want to look at dorsolateral prefrontal cortex, for instance, is involved with many different kinds of functions. We're interested, obviously, in functions that have something to do with emotional regulation in this case. And emotional regulation has a long history to it. And in terms of neuroscience, it goes all the way back to approach and avoidance behavior that have become positive and negative valence systems built into the mammalian brain. And at the cortical level, we're working with a collaborator named Tim Strauman at Duke University, who has created a neurobiological theory that have to do with, in terms of approach and avoidance systems, has to do with promotion goals and prevention goals at a cortical level in terms of how we regulate our emotions, in terms of our interaction in the world with people. And when these systems go bad, he theorizes that that has something to do with how depression, for instance, begins to occur, and how anxiety symptoms also occur in humans. And so we actually have some very specific functional networks associated with depression and anxiety that we can aim at in the brain. And the other part of that is, well, we need a way to activate that in the brain so we can individually target where to go. I mean, for instance, dorsolateral prefrontal cortex is a big place. And what you really want is the specific areas in the brain that have something to do with emotional regulation. And so Dr. Strauman has already developed tasks that light up dorsolateral prefrontal cortex as one place, but at other places as well in these depression networks. The task that he uses is a priming task where words that are associated with your personal goals, we get them from interviews ahead of time, both prevention and promotion, are used. They're actually only flashed on the screen for a very, very short period of time. The subject can't actually even read them. The subject's actually just looking for a red characters, and they're supposed to hit the button just whenever that happens. But the brain actually can read these words. They're only up there for 30 or 40 milliseconds, but they're up long enough for the brain to read them and light up these actual depression networks, including dorsal prefrontal cortex. And it turns out that in a healthy volunteer, you see these networks lighting up, say this particular part of dorsal prefrontal cortex, whereas people that are depressed do not. That's this green line. So what you would want to do on the left side of the head is somehow normalize a depressed person's activation in this task, for instance. In some sense, that may mean that you are working on this network, you're modulating it, you're changing it in a way that may help with the depression. And so, very interestingly, what you want to do with TMS is to use excitatory TMS on that side of the head in that area, which is exactly what is being done. That's the FDA-approved stimulation, which is to use 10 hertz stimulation, which is excitatory, in left dorsal prefrontal cortex to upregulate cortical excitability. And it's just that now we're able to do this on an individual basis, targeting exact spots, okay, in the brain where we should actually aim the TMS. So we now have that idea that you can use functional targeting, okay, in the brain using this task, okay, to find the area, and then how do we actually do the CPAS approach? Well, we give a certain therapy, okay, for instance, cognitive behavioral therapy, that actually activates the same areas in the brain and have been shown to change in the brain over a course of treatment. Okay, so the cognitive behavioral therapy actually is one example of an area that changes dorsal prefrontal cortex. So it's the same place as that task lights up the stimulation. So we're able to find an individual target using TMS, and then we will give people the normal FDA-approved TMS, namely daily 10 hertz stimulation, but we're also having them have therapy, in this case cognitive behavioral therapy or other kinds of therapy, talk therapy as well, at the same time concurrently with the TMS. That's the functional targeting aspect of this. And so we actually did a pilot study with just 5 people, open label, at Duke a year or 2 ago, with 5 treatment-resistant depressed patients. And we gave them 4 weeks. We could give them 6 weeks. That's the normal FDA-approved amount. But we gave them 4 weeks of simultaneous TMS while they were getting the cognitive behavioral therapy, which in this case is called self-system therapy. And what we found is that all 5 people remitted. All 5 people got better. Now, this is open label, and it's only 5 people, so that's encouraging, but it's not proof of anything. But like I said, it is encouraging. The very interesting thing that we found, and we found this in 2 different ways, and this is only, again, 5 people, but we saw both in univariate analyses that areas of the brain in the hippocampus and in the ventromedial prefrontal cortex, which are areas that are connected to dorsal prefrontal cortex, are part of these networks, and areas involved with depression, were both affected by the TMS, the course of treatments, looking at it pre and post. And they actually increased in their activity. And we were actually able to show using graph theoretical analysis as well that prefrontal cortex increased its connectivity in these 5 people, all 5 people, with temporal lobe areas that are also have to do with which, for instance, the hippocampus, which also have to do with depression. So we were able to show network changes in just 5 people, all 5 of them, that have to do with changes in network activity associated with depression. And the fact that in an associated way that they all got better is highly, highly encouraging. And so what we did what we're saying then is that the CPAS approach, right, the idea of engaging specific brain networks associated with a targeted psychological function, in this case, those that are associated with emotional regulation and depression, using functional neuroimaging, and then using non-invasive brain stimulation, in this case TMS, and functional targeting, concurrent therapy at the same time as the TMS is given. These are all means of increasing the efficacy of the treatment that we're trying to do, and really gives a very strong scientific basis to what we're doing with TMS in depression. On that note, we've begun here at the NIMH, we've begun a larger clinical trial to test this whole approach. Like I said, we had a nice pilot that encouraged us, but now we're trying to, we're using active and sham TMS to be able to really decide, and looking at network changes under both conditions to see, if what we're saying, has the validity we think that it does. So we're actively engaging eligible adults who have major depressive disorder, and even people that are still taking antidepressants. It doesn't matter whether you're on or off. It's also inpatient or outpatient, depending on where you live. It lasts for 8 weeks. It's a 6-week period of TMS with testing on both ends of it, and then we do have some follow-ups after that. And I have some little addresses there, or a phone, or if you're interested, we would love to talk to you. And these are just in terms of acknowledgments. These are some of the many people that we work with. This is work that I've been involved with, especially with Holly Lisenby, and now with Carlos Zarate at the NIMH for many years, and people at Columbia and at Duke that we've been working with for many years as well.